Hello everybody, welcome to Azan Explains IB. Today we are looking at Unit 1.1, Understanding Statement 1. The statement of today states that, according to the cell theory, living organisms are composed of cells. We are going to be approaching this statement today and trying to dissect it and learn as much as we need to regarding the cell theory and regarding the statement. Okay, so the statement says that according to the cell theory, living organisms are composed of cells. So when we break this down, we need to know what the cell theory is. So the cell theory is composed of three statements. It is the combination of these three statements, and the statements are: all living things are made of cells. Cells are the simplest units of life, and all cells come from pre-existing cells. We need to understand as well as to how these statements came about, and when we look further into Unit 1, we'll start delving into Statement 3 a lot more closely. So Statement 1 came from Matthias Sheldon and Theodore Swan when they both were. So Matthias Sheldon was a botanist while Theodore Swan was a zoologist, and they both realized that under microscopes, which were becoming a very big thing back then, they found that all plants and all animals contained cells. And they finally found out after much communication, a lot of communication, that all plants were made of cells and all animals were made of cells. Hence, all living things must be made of cells. That's the first statement. The second statement says that cells are the simplest units of life. How do we know this? Well, it's because Robert Hooke, who was the first person to coin the term cells when he looked at a cork, and cork is nothing but dead plant cells. So when he looked under a cork in a microscope, he found that all he saw were little rooms within the structure of the cork. And they represented something that of the same cells that monks used to pray and worship and meditate in. And so he referred to those tiny little rooms as cells. But what's interesting about what Robert Hood looked at is that everything was dead. A cork is nothing but dead plant cells. So he only saw the cell wall of the cork. However, at the same time, Leeuwenhoek, who was one of the key figures to create the microscope, he looked at alive cells, and he did this by going into ponds and finding organisms there. He found within the whatever mess was within the mouths of people who couldn't brush their teeth, because hygiene was not as great back then. He also found that there were living organisms within within these substances, and what he found was that um, with Leeuwenhoek's discovery, they had organelles, and organelles are what make the basic organization of a cell. So it came to be that if Robert Hooke was looking at alive cells and Robert Hooke was looking at dead organisms and Leeuwenhoek was looking at alive organisms, it also means that cells would be considered the simplest unit of life as there are, besides its organelles, which essentially make up the cell. There is nothing which is any um, nothing can be smaller than a cell and its organelles that can be considered alive. And so it is the simplest unit of life. Finally, the third statement: all cells come from pre-existing cells, and this states that cells must have come from cells before it, and this refers to the process of mitosis and how daughter cells are born through reproduction. It was found through two people, Rudolf Virchow, who conducted autopsies and conducted the germ theory. However, it didn't contain a lot of data and it wasn't exactly very backed up. However, there was an experiment conducted by someone called Louis Pasteur, which, if you may or may not have noticed, also refers to the same scientist who discovered who created pasteurization, which saves millions of lives because of clean milk, which is safe to drink. And so he conducted what is known as the Swan neck flask experiment, whereby he got something which is known as so he did, this Swan neck flask experiment is also known as Pasteur's experiment, and it revolved around using a Swan flask. Now, my drawing isn't exactly very good, but this pretty much gets the idea across that the Swan neck was particularly designed so that the groove within the neck of the flask, so if I were to focus on this part, the groove on the neck of the flask made it so that substances which entered through here would often get stuck here because of the way the flask was shaped. So using this idea, what he did was he conducted two series of experiments. So this is experiment 1 and this is experiment 2. So his independent variable was that the Swan neck flasks in experiment 1 would stay attached. So the Swan neck itself was still connected to the flask. And in experiment 2, however, he actually broke the flask. He broke the Swan neck flask so that only the flask side of it, which contained the microbial broth, which is essentially a way of, it was a way of promoting cell growth because it had all the nutrients and conditions required. What he found was that when the neck of the Swan neck flask was maintained and was never broken, it stayed clear. The solution never changed color, and hence it showed that after introducing the dust and microbes and finding that the solution was clear, it was because the dust which got introduced into the flask got stuck in the Swan flask, which meant that there was no microbial growth, and they could not pass due to gravity and the way the neck was designed to trap all dust and microbes. However, with experiment 2, what he found was that when he broke the neck of the flask, it exposed the nutrient broth, and after introducing the dust and the microbes, the microbial growth started to occur. So he started seeing that the solution was turning very cloudy, and he found that it was showing microbial growth after analysis as well. Why though? Because when the dust was introduced, it carried microbes. And as the microbes got into the solution, because there was no Swan neck to restrict it, the solution and it entered into the solution and became cloudy. This is all well and good, but really, what does this mean? Well, it shows that cells must have come from pre-existing cells. How so? Well, because if the cells came from spontaneous generation, which is at the time believed to be a way of that cells would originate simply by themselves. They just appeared from nowhere and they would just appear. And while this doesn't make sense to us now because we clearly know that this is false, back then it was considered a very large ideology. So here it says that if cells came from spontaneous generation, then the microbial growth would have come from both flasks. This flask which stayed clear, would have also turned cloudy. Because if it truly came from spontaneous generation, then the cells would have also been able to generate themselves through the microbial conditions which are favorable for growth. But they didn't. Instead, it only occurred when dust entered into the solution, which contained microbes, pre-existing microbes, and became cloudy, hence proving that it can only occur due to um, it shows that cells can only have come from pre-existing cells. This experiment will prove very important, as I mentioned previously, in Unit 1.5 when we study endosymbiotic theory, because you will need to understand a little bit more regarding the origin of cells themselves. For now, though, this is Unit 1.1, Understanding Statement 1. Thank you for watching.